The title of this talk is Introduction to Drug Metabolism. The learning outcomes are: what is drug metabolism? What is the importance of drug metabolism in drug discovery and development? What is the influence of some physicochemical properties on drug metabolism outcomes? The panel below shows an example of a drug being metabolized by cytochrome P453A4, in this case, buspirone being oxidized to hydroxybuspirone. We will see more of these kind of examples as the talk progresses. But metabolism is the enzymatic conversion of a compound to a more water-soluble form. Metabolism is the enzymatic modification of compounds by increasing their water solubility and therefore their renal and biliary excretion. It is a determinant of oral bioavailability, clearance, and half-life in vivo. Metabolism occurs predominantly in the liver, but other organs such as the intestine, kidneys, lungs, and skin are capable of metabolizing drugs as well. The image to your right of the slide shows you the relative importance of these organs to drug metabolism. The most important organ is the liver, followed by the intestine, the kidneys, the lungs, and the skin. Metabolic stability can be improved by structural modification, which blocks or sterically interfere with metabolic sites. We will look at this in more detail later on. The relationship between drug metabolism, pharmacokinetics, and pharmacodynamics. The relationship between drug metabolism, pharmacokinetics, and pharmacodynamics can be looked at as a continuum where drug metabolism drives pharmacokinetics and pharmacokinetics in turn drives pharmacodynamics. So they are interrelated and they influence each other. Introduction to phase 1 and phase 2 metabolism. Phase 1 of functionalization reaction exists to produce or uncover functional group for phase 2 by oxidation, reduction, or hydrolysis. In other words, it prepares compounds for phase 2. Phase 2 or conjugative reaction takes products from phase 1 and they are conjugated by phase 2 enzymes such as uridine diphosphoglucuronal transferase, UGT, or sulfur transferase. This process generally makes them more water-soluble and therefore more readily excretable in bile or urine. These two processes, phase 1 and phase 2, are important in the drug discovery process as they are responsible for metabolizing various drugs. Metabolic reaction is a chemical process where it could be by oxidation, in this case looking at the aliphatic oxidation of propanol by alcohol dehydrogenase to propaldehyde, which undergoes one-step oxidation further to give us propionic acid. So we're taking alcohol and making it into an acid by two-step oxidation by indeed two different enzymes. We can have reduction, and for this example we are looking at the reduction of nitrobenzene by nitroreductase to aniline. We could have hydrolysis where water is added to break indeed an ester bond or an amide bond in examples. We could have conjugation, which is essentially a condensation reaction in phase 2, In this case, we're looking at the glucuronidation of catechol to catechol glucuronide by UGT. General rules of metabolic reaction. The liver is the major site, although several other organs can play an important role. Compounds are often converted to hydrophilic derivatives, but there are exceptions, as in the metabolism of aniline, where aniline is converted to methyl-aniline or by acetylation to acetanilide. In both of these cases, the product methyl-aniline and acetanilide are less water-soluble than the aniline from which they were derived from. Metabolism often renders drug pharmacologically inactive, but there are exceptions to this as well, as in the case of imipramine and desipramine, which are both antidepressants, but desipramine is a better antidepressant than the imipramine from which it was made from by N-dimethylase. Metabolites are more readily eliminated than parent drug, not always, because we have examples where the metabolites are longer acting and indeed long half-life than the parents from which they were started from, as in the case of morphine 6-glucuronide. And we should bear in mind that some compounds are not metabolized at all. Metabolism of benzene acts as an example of metabolic reactions. Benzene is a non-polar moiety which can be made more polar progressively by oxidation and further oxidation. So benzene can be oxidated to phenol, which is more water soluble, and this can be further oxidated to give us the benzaldehyde, which can undergo further oxidation to give us the benzoic acid. The benzoic acid can undergo one of two reactions. It can be glucuronidated to give us the benzoic glucuronal conjugate, or it can undergo glycination to give us epuric acid. The phenol that is formed from benzene can undergo sulfation to give us the phenolic sulfur conjugate, or it can undergo glucuronidation to give us phenolic glucuronal conjugate. Or it could also undergo methylation to give us the methoxybenzene. But the benzoic acid so formed from phase 1 can undergo glycination to give us epuric acid. What we can see from these many reactions is from one simple compound, benzene, we can form several metabolites of differing polarity as the reaction proceeds. So this example shows how a non-polar compound can be made more polar and therefore more readily excretable in urine or bowel. This next example is that of the metabolism of verapamil. Verapamil is a calcium channel blocker which is used for controlling hypertension. Verapamil can undergo several reactions. It can undergo O or N demethylation to give us the product to the left. This can also undergo N-dealkylation by CYP1A2 or CYP3A4 to give us the dealkylated product. It can undergo N-demethylation by CYP3A4 to give us the demethylated product, or O-demethylation to give us several O-demethylated products. It can undergo O or N-glucuronidation also to give us the phase 2 metabolites. Verapamil has more than 30 different metabolites which are still being elucidated even till today. Some more metabolites are coming to be known. Metabolism and pharmacological outcomes. The intensity and duration of action is related to the concentration of drug at active site and time at the active site. So rapidly metabolized drug will have short duration of action. Pharmacological activation. Metabolite has greater affinity than a parent drug for the receptor. In this case, the example of codeine being converted to morphine and then further on to morphine 6-glucuronide, which is more than 100 times more potent as an opioid drug in the CNS. The conversion is shown as in the panel below, where codeine is being O-demethylated by CYP2D6 to morphine. We could have pharmacological deactivation where a metabolite has lower affinity for the receptor, which generally the case is what happened with phase 2 conjugations, which could be classified as a deactivating process. We could have a pharmacological change where the metabolite interacts with a different set of receptor from that which the parent interacts with. CYP450 is capable of metabolic switching. This can be advantageous when there is likely to be a drug-drug interaction by the blockage of one metabolic process by another drug. Metabolic switching occurs when more than one CYP isoforms can catalyze the same reaction. In this case, the N-dealkylation of verapamil again, which can be performed by CYP1A2 or CYP3A4. So if CYP1A2 was blocked, CYP3A4 will assume the sole role of N-dealkylation of verapamil and therefore preventing buildup or drug-drug interaction. CYP3A4 can also undergo drug-drug interaction, and other isoforms are capable of experiencing or undergoing drug-drug interaction as well. Drug-drug interactions occurs if two or more drugs are co-administered and one drug inhibits or induces the metabolism of a second drug at a particular enzyme or isozyme, leading to toxic effect or loss of pharmacological activity. In this example, we're looking at nevirapine, which is an antiretroviral drug metabolized by CYP3A4, and rifampicin, which is an antitubercular drug which can cause the induction of CYP3A4. So if we have a patient that is comorbid with HIV and tuberculosis, the idea then becomes these two drugs, because rifampicin induces the metabolism of nevirapine. Nevirapine will not be as active against HIV as it could be, so it may necessitate an increase of the dosimetry such that there'll be enough nevirapine to still be able to combat the HIV or vice versa. So generally, in this case, this is one classic where drug-drug interaction can lead to inadequacy of therapy, in this case for antiretrovirals, but this can be mitigated by increasing the nevirapine dose or decreasing the rifampicin dose, whichever one is deemed ideal by the clinician in charge. There are other phase 1 and phase 2 enzymes. The other phase 1 enzymes are flavin monooxidase, FMO, alcohol dehydrogenase, AD, aldehyde dehydrogenases, ALDH, of different isoforms, monoamine oxidase, MAOs, B and A, xanthine oxidase, aldehyde oxidase. nitroreductase, azoreductase, and of course esterases and amidases. A nitroreductase enzyme can convert, in this case, nitrobenzene to aniline. There are other phase 2 enzymes just as well, apart from UGT. There are sulfotransferase, methyltransferase, N-acetyltransferase, catechol O-methyltransferase, acyltransferase, phosphotransferase, and glutathione sulfotransferase. In this panel to the right, we're looking at the conversion of catechol by catechol O-methyltransferase to methoxycatechol. Some of the other phase 1 and phase 2 enzymes are named after the functional group that they catalyze. Test systems for assaying metabolic stability. There are different test systems for investigating compound stability, such as liver homogenate, which is just an homogenate of liver in pH 7.4 buffer. There's liver S9, which is a supernatant from the 9,000 times G centrifugation of the liver homogenate. This contains the unwashed cytosol and microsomes and can be useful in situations where there is no access to an ultracentrifuge in the lab. Then we have the liver cytosol, which is a supernatant from the 100,000 times G centrifugation. The cytosol generally contains soluble enzymes, such as aldehyde oxidase, for example, in this case. We have liver microsomes, which is the wash pellet from the 100,000 times G centrifugation, which contains primarily P450s and other membrane-bound enzymes such as FMO and UGT. And finally, we have the hepatocyte, which is the intact liver cell, which has all possible metabolic capabilities without the requirement for a cofactor. Test system for assaying metabolic stability. Microsomes can be used to perform both phase 1 and phase 2 reactions, but requires the addition of specific cofactors such as nicotine adenine dinucleotide phosphate in its reduced form, NADPH with or without uridine diphosphoglyceronic acid, or phosphoadenosyl phosphosulfate for sulfation reaction. Cytosolic enzymes are capable of both phase 1 and phase 2 reactions, but some may require the addition of specific cofactor as well. The hepatocytes, as already alluded to, can be used to perform the full range of integrated hepatic metabolism without the need for cofactors. The schematic to the right of your slide shows the interplay between microsomes and hepatocytes. Microsome, which is part of the hepatocytes, contains just mainly 3 membrane-bound enzymes, cytochrome P450, UGT, and FMO, whereas the hepatocyte contains all the enzymes that our drugs could ever be exposed to. The typical unit of intrinsic clearance, which can be expressed in either microliters per min per mg of microsomal protein or mils per min per gram of liver. We generally want this, if it's the uncorrected version, to be less than 95.2 microliters per min per mg of microsomal protein, or less than 5 mils per min per gram of liver. The lower these numbers are, the more stable the compound is. Physicochemical properties. Lipophilicity is an important physicochemical parameter influencing processes such as permeability, solubility, oral absorption, brain uptake, free circulating concentration, and various in vivo pharmacokinetic properties. Increasing the lipophilicity of a compound can increase its oral absorption. It can increase the extent of its plasma protein binding and the volume of distribution. However, the more lipophilic a compound becomes, the more vulnerable it can be to cytochrome P450 metabolism, which prefers to metabolize lipophilic compound and therefore giving us high compound clearance. The molecular size and hydrogen bonding capacity are two other properties that are often considered as important for membrane permeation and pharmacokinetics. And a balance of hydrogen bonding for activity with the target as well as with the enzyme is judiciously to be chosen because the hydrogen bonding is required to engage with the target, and equally it can help engage with the enzyme. So a trade-off between the two is required. A general guide for lipophilicity. A compound that has a log D 7.4 of less than 1 can indeed demonstrate good solubility but low absorption and brain penetration owing to slow passive permeability. It can have high clearance by kidney owing to their polarity and may exhibit paracellular permeability if molecular weight is low. The ideal range for log D 7.4 is between 1 and 3, and compounds in this range generally have good intestinal absorption owing to good balance of solubility and passive permeability. Metabolism is minimized owing to low binding to metabolic enzymes. Compounds which have a lipophilicity of between 3 and 5 can have good permeability but poor or lower absorption owing to lower solubility. Metabolism can be increased in this range owing to increased binding to metabolic enzymes. And lastly, compounds which have log D 7.4 greater than 5 can have low absorption and bioavailability owing to low solubility. They can have high metabolic clearance due to the high affinity of the drug for the metabolic enzymes. The volume of distribution and half-life could be high because of compound partition into tissues wherein it might stay. Successful strategies for increasing metabolic stability. We can block metabolically labeled site by the addition of fluorine or other halides. We can block the metabolic site by the addition of other groups. We can remove labeled functional group. We can cyclicize the compound and make them more rigid so that they are not easily metabolized. We can change the ring size from a 5-membered ring to a 6-membered ring or vice versa. We can change the chirality of the chemical compound. We can reduce the lipophilicity, and we can replace unstable groups. A word of caution that the optimization of molecules to remove liabilities and to retain activity often involves contradictory changes to the properties of the molecule. Drug molecules are often finely balanced, compromised between these properties so that you get some gain in potency and maybe some gain in stability, but minimizing the changes that can happen to potency. Use of blocking groups. In this example, we are further following up on the metabolism of buspirone as earlier indicated. Generally, addition of blocking groups such as fluorine to prevent oxidation of the phenyl ring, which usually occurs in the prior position, can be helpful. In this case, we are looking at the blocking the site of oxidation with a fluorine in this case. So we have blocked the buspirone such that it's fluorobuspirone, But when we look at the resultant metabolic instability, we can see that the metabolism by CYP3A4, the half-life has gone up from 4.6 minutes to 52.3 minutes for the fluorine-blocked buspirone. But equally, we've had about twofold loss in potency. But in this case, the loss in potency is inconsequential because it's still in the nanomolar range. So you can see that you may gain indeed stability and lose some ground on potency or vice versa. So it's a judicious use of this information that will be helpful in propelling the compounds forward towards candidacy. In conclusion, we have learned that drug metabolism is an enzymic process rather than a spontaneous process. That there are two different phases of drug metabolism reaction, namely phase 1 and phase 2. The enzymes involved in each phase have also been covered, the chief of which is cytochrome P450 for phase 1 and UGT for phase 2. We've looked at the influence of some physicochemical properties, primarily lipophilicity, on drug metabolism. We've investigated the different test systems that are available for investigating drug metabolism, such as liver S9, microsome cytosol, and hepatocyte. We've also learned how to mitigate rapid metabolism by use of blocking groups to block the metabolism hotspot on the compound. Thank you for your attention.